No, thanks for checking in though. Okay, it's 5:20, so we leave the door open for just a couple of minutes to have the rest of us join. But for the rest of you, hello, welcome to week 2 of our course on nutrition and cardiometabolic diseases. This week, we will be talking all about the pathophysiology of type 2 diabetes. And this was something that I know came up in little bits. There was some interest in type 2 diabetes while we were talking about the introductory lecture from Mariana last week. So Iris Van Damme, who is my colleague and collaborator at the Division of Human Nutrition and Health, will be leading all of our plenary sessions, all of our lecture series for this week. Iris is a fantastic PhD candidate in our chair group, Nutrition and Disease. She is supervised by Mariana. Her focus and her topic of research is nutrition interventions in type 2 diabetes, more or less, broadly. Yeah, she's going to tell you a lot about that subject over the course of the next 4 or 5 days, but also a little bit about herself. Before we get started, Iris, is it okay if I just spend a few minutes with all of you to just check in to see how everyone is doing with your logistics for group work, getting in and getting to know your group members? Has that been okay? Has that process started? I see some people nodding. Thank you very much for the groups who have indicated their preference for when they would like to meet during in-person group work time. There are 3 groups that I have not heard from yet. Would you be able to let me know by the end of today what your preference is? It would just be handy for us to organize our group sessions that way. I'm assuming the groups that did write to me, they're thinking not just for this week, but also their preference for the entire course, right? That's my understanding of it. So whatever time you have chosen, I'm going to assume that that's what works best for your group for to the end of December. So that's great. So the rest of you, could you please just let me know when what your preference is to meet in your group? That would be great. The idea again for that is just to make sure that we organize the spaces and give you time to separately discuss within your group, but also come collectively so that you can share your questions between each other. Also, if you have questions for us, we can respond to your questions, and other groups who may or may not have thought of your question would learn from our responses. So there's a little bit of that we'd like to organize. Besides that, there are self-paced online courses, SPOC modules. I hope you have all had a chance to register and have played around with that, both in the previous week and this week. So you have some modules to follow there. I know those courses are now functioning. They started last week. So that was just a quick reminder for you. I'm just checking my list to make sure that everything else, I think that was pretty much all of the housekeeping announcements that I had for all of you. Is there anything else that you'd like to quickly chat about or discuss? Just slides for this week. We had thought of sharing on Brightspace after the course, like after the session. So after Iris finishes the session, we post the slides. I know this might be a little inconvenient, but the reason is we're trying to facilitate group discussion, and some of the answers are in the slide deck, so we don't want to give those to you ahead of time if possible, which is why we're sort of holding out to after. Just so you know, they will be posted, but they're now going to be posted after the lecture instead of before. Any other aspects? No? Okay, then we can get started. Just know I will be here in the break or after if you need to reach out to me with any other questions that you can't think of right now. All right, so then let's get started. Iris, would you like to? Yeah, just make sure we're recording.
Recording?
Yes, I think so.
Oh, here we go. Okay.
Yeah?
Yes,
Thanks very much.
Thanks for the
I just
introduction.
put this down.
As Nea already told you, my name is Iris. I'm working as a PhD candidate at the Division of Human Nutrition. And indeed, I'm doing research into the field of diets for people with type 2 diabetes. Actually, what I'm investigating is how we can lower the risk of cardiovascular diseases for people with type 2 diabetes, which I think is very relevant for this course. So I will, throughout this week, tell you more about my research, but also, of course, about a lot of other studies that have been done in the past. So for today, we will mainly talk about the pathophysiology of diabetes, and to share a bit more about what we will do the rest of the week. I made this schedule. You can see that tomorrow we will discuss more about diets and diabetes, so that's more focused on nutrition. And then on Thursday we have another lecture scheduled. And in this lecture I would like to discuss with you the assignment that is scheduled for this week. So there's an assignment which is about some calculations with insulin resistance, insulin sensitivity. And I think you can work on that alone or together, whatever you prefer, in the afternoon of Thursday. And then in the end of the afternoon we will just discuss with everyone as a group what the answers will be, or if you have any questions, you can ask it then. We also ask you to hand in this assignment. You can do this via Brightspace before 5 o'clock on Friday in the afternoon. Just for the entire lecture, if you have any questions or you have anything just you want to share, just raise your hand and I can answer your questions. I'm not sure if there are any questions now already, but if not, we will move on. I split the lecture of today in two parts, so we have a break in between. In the first part I will mainly talk about symptoms and the prevalence of diabetes, and a bit about the measurements of glucose and insulin sensitivity. And then after the break I will tell you more about risk factors for type 2 diabetes and also a bit about the treatment options for the disease. So to start with the symptoms and treatments, this figure shows actually the relation between cardiovascular diseases and diabetes. On the left side you see in blue the people who have type 1 diabetes, and on the right side people with type 2 diabetes in blue. And it shows actually the number of people that die from any form of cardiovascular disease. In red you see matched control, so those are people who do not have diabetes but are very similar to the people who have diabetes, and you clearly see that people with diabetes, either type 1 or type 2, have a higher risk of dying from any cardiovascular disease. And throughout this lecture I would like to explain you why this actually is the case, and also maybe what we can do about that. But before that, I think it's good that we agree on what we are talking about actually. So I refer to diabetes, but the official name is actually diabetes mellitus. It's a Latin phrase, and if we translate this, it means something like passing through honey sweet, which relates actually to the urine, because years ago people with diabetes were diagnosed by the fact that their urine was very sweet, so doctors really drank people's urine to see if they really had diabetes, and if it was really sweet, they thought, well, you probably have diabetes. Because people with diabetes have a lot of glucose in their body, and they also have a lot of glucose that they get rid of via their urine. But nowadays we know that it's not only the sugar in the urine that is very typical for diabetes, but it's also the sugar that is in your blood. So diabetes mellitus is a serious disease that is characterized by high levels of glucose inside your bloodstream, and this can be due to two different reasons. So the first one is that your body isn't able to produce sufficient levels of insulin. The second reason is that your body is able to produce insulin, but that it doesn't respond well to this hormone. And sometimes it's even a combination of the two. But both these mechanisms, so either being insulin deficient, not being able to produce sufficient insulin, or being insulin resistant, not being able to respond to this hormone, leads to high levels of glucose in your blood, or in other words, hyperglycemia. What you see here in this picture is what happens when people eat something, and normally your glucose levels, it's the blue line over here, normally your glucose levels start to rise a bit after a meal, and then they decrease over a really short period of time. But when you have diabetes, these levels are really high after you had a meal, and they also stay high for a very long period of time. And of course, there's some gray area in between, where you see here mentions pre-diabetic, so that's actually a state where you don't have diabetes, so you don't fulfill the criteria of having diabetes, but you already have elevated glucose levels. I also said it's a serious disease, and I think this picture really shows that it's indeed a serious disease, as there are about 11% of the global population has some form of diabetes. You see on the left side that this prevalence rate increased over the last 20 years, and on the right side you see the expectations for the coming 20 years. You see that the numbers are even increasing further. So there are a lot of people having diabetes, not only here in the Netherlands, but worldwide. It's a really prevalent disease. And about 90% of the people with any form of diabetes has diabetes type 2, which is the most common form of diabetes. But there are more types of this disease. So you probably heard before type 1 and type 2 diabetes, and maybe also a diabetes that occurs during pregnancy. But there are actually more types as well. So here you see the three most common forms, actually. So on the left side you have type 1 diabetes, which is mainly characterized by an insulin deficiency, which means that the body isn't able to produce the insulin. And then type 2 diabetes is actually a combination of the body being not able to respond to the hormone and also not producing sufficient levels of insulin. And the same is the case for gestational diabetes. But as I said, there are more types, so here I put down some more information. For example, that type 1 diabetes is an autoimmune disease, which means that the body of people with type 1 diabetes kind of attacks the cells that produce insulin, and that's why they aren't able to produce sufficient levels. But there are also other types, such as MODY and LADA. MODY stands for maturity onset diabetes of the young. So it's actually a genetic disease, and it usually develops, it's kind of similar to diabetes type 2, but then it already shows up early in life, because we always tend to say that the type 1 diabetes is something that is diagnosed in children, and type 2 diabetes in adults, because it takes a longer time to develop. But apparently something is happening in the people with this genetic variant, that they have the same similarities to type 2 diabetes, but then it's already diagnosed when you're really young. And we also have LADA, which is a latent autoimmune form of diabetes. So in this case, people have an autoimmune disease, but this does not show up when they are young, but it develops over a longer period of time, which means that they're, you can only diagnose the disease when they're adults. As Nea also said in the beginning, I'm mainly doing research in the field of type 2 diabetes, and I also think that in respect to this course of cardiometabolic diseases, type 2 diabetes is the most common form, and so most of the rest of my slides will also be about this type of diabetes. But of course, if you have any questions about the other types, feel free to ask. There are also other types or other ways of classifying people with diabetes. And I think this paper, which is done by a group of Scandinavian researchers, really shows that, our traditional clustering of people with type 1 and type 2 diabetes might be a bit odd, I would say, because here in the picture above, or picture A, you see if you group people with diabetes into the classical type 1 and type 2 diabetes, and here they also show the people with LADA, so the latent autoimmune form of diabetes. You see that the vast majority will classify as type 2 diabetes, 93.6% in this case. But when you look in figure B, here, you see they made different groups which actually divides the people with type 2 diabetes into smaller clusters. And what they did here is they checked actually the cause of their high glucose levels, so why have these people hyperglycemia? And what they saw was that some people have high blood sugar levels because they are just getting older and something is happening in their body leading to hyperglycemia. Other people are obese, and that might be the cause of their high glucose levels in the blood. Whereas other people are clearly insulin deficient, so although they are classified as people as type 2 diabetes, their bodies aren't able to produce sufficient levels of insulin. And there's a group of people who are mainly classified as insulin-resistant diabetes. And this picture actually shows that there are multiple ways of classifying people. I think it's very important also if we want to improve treatments for people with diabetes, that it's important that we look into the cause of this. Whereas you can see in the picture above that if we say all people with type 2 diabetes are the same, that might not be the best option to treat them. So to move on, I would like to show you the symptoms of diabetes, and in this picture you see both, actually symptoms for all types of diabetes, and in red the symptoms that are more common for people with type 1 diabetes. You see here it still refers to something with urine or the kidneys here, so it says polyuria, which means people to go to the toilet very often, so they have frequent urination. And also glycosuria, which means there are still a lot of glucose present in their urine. And that also refers back to the way diabetes was diagnosed years ago. But there are also other symptoms, such as people have a lot of thirst, they can be very hungry, they can have blurred vision, so there can be vision problems. But also things like weight loss or being nauseous. They have sometimes a typical smell that smells like acetone, or typical breath, sorry. So there are a lot of symptoms that, could be due to having diabetes, and some of these are more specific for people with type 1 diabetes, but sometimes it's a bit, I would say vague what kind of symptoms people will have. So you may ask, how do we then diagnose diabetes? Well, for that we have clear cutoff values. Here, those are from the WHO, and what we diagnose people based on the level of sugar or glucose in their blood. So there are different ways you can do this. For example, by measuring fasting plasma glucose levels, so people stay fasted for a night, for example, for 8 to 10 hours, and then you measure how much glucose is present in their blood. And in a normal case, it should be relatively low, so below the value of 6.1. But if it's higher, for example, higher than 7.1, you see people will classify as having diabetes. And these diagnostic criteria are both the same for people with type 1, type 2, any form of diabetes. There's also something here with 2-hour plasma glucose levels, and this reflects to when people are not fasted, but they did eat something, and then you measure their glucose levels 2 hours after. But also there you see when the levels are elevated, people will classify as having diabetes. So if they're higher than 11.1, we will say that people do have diabetes. Then there's also this measure here. It says HbA1c, and also I will explain this a bit in more detail later on. But also for this criteria, you see that the higher the value, the higher the risk of diabetes, I would say. So in between, there are two other, I wouldn't say it's disease, but we refer to it as pre-diabetes. So you have here impaired fasting glucose and impaired glucose tolerance. And these are, as I said, we refer to them as pre-diabetes. So it's some sort of pre-stage before people have diabetes. We already see that their glucose levels are elevated. And this means that people who fulfill in the middle category, so these two, have a very high risk of progressing to having diabetes. Yes?
Yes, can I just confirm, is the diagnostic criteria, the screening criteria, any one of these, or is it a combination of all of them?
No, it's any one of these. So in the Netherlands, I'm not sure if it's the same in all countries, but in the Netherlands, what doctors usually do is they measure fasting plasma glucose levels they tend to do it once, and if they see the levels are really high, they might do another check, or they are, well, if they are already very high, people are often diagnosed with diabetes. Yeah. I also put down here 2 different values for the HbA1c, because previously we mentioned this value is a percentage, but since a couple of years we changed to millimole per mole. So this is actually the new diagnostic criteria. But in some papers or some research from a couple of years ago, you will still see the percentage value. So I think it's good also to know that these exist as well. But also for the percentages, it's the same. The higher risk of diabetes. And to tell you more about this HbA1c, it says here it's the part of hemoglobin that is glycated. And to tell you more about that, in your body you have red blood cells that travel throughout your body, and there's hemoglobin inside these red blood cells. And this hemoglobin can bind glucose. So if glucose is traveling through your veins because you ate something and you're going to get the glucose of your meal, it will circulate inside your body, and it can bind to the hemoglobin. And there it will stay. And this HbA1c actually reflects how much of the hemoglobin that is present in your body is attached to a glucose molecule. And because red blood cells they usually stay alive for 2 to 3 months, so you can kind of look back in time and see how much glucose was bind to the hemoglobin in your red blood cells for the past 2 to 3 months. So it's actually kind of an average value for your glucose levels over a longer period of time, and that is why we like to have this value as well next to the fasting plasma glucose levels, because as I just said, you can measure your fasting glucose levels just once, but it might be more reliable to have this HbA1c level as well. But in the Netherlands, at the moment we don't use this as the first way of diagnosing people. We still refer to the fasting glucose levels. This HbA1c also has a clear relationship with cardiovascular disease. So what you see here, I hope it's also visible in the back. You see white squares and blue circles. The white squares are the number of people with myocardial infarction, and in blue the microvascular endpoint, so it's also a form of cardiovascular disease. And you see that the higher your HbA1c, the higher your risk of any cardiovascular disease in this case. But it also shows a clear relation between people who have diabetes, because they're of course on the high level of the HbA1c and the high incidence of CVD. And to highlight this even further, I think Marianne last week also told you a bit about the Framingham study or the Framingham score. In this study, they already found that people with diabetes have a very high risk of getting cardiovascular disease, and that they even have a 2 to 4 fold higher risk of cardiovascular mortality. So it's not only that they have high risk of getting cardiovascular disease, but also they have a high risk of dying from cardiovascular diseases. There are also a lot of other complications that can occur in people who have diabetes. Here in this picture, they make the distinction between microvascular and macrovascular and other types of complications. So what people with diabetes often, also I said the blurred vision is a typical symptom of having diabetes. That's because there's some damage in your arteries in your eye, which makes it difficult to see. And we refer to that as diabetic retinopathy, so there's something in your retina that is damaged. But there are also other microvascular complications, such as problems with the kidney or problems with your nerves. And on the right side, we have the macrovascular problems, which refer to the cardiovascular diseases. So you can have problems in your limbs, but also in your heart or in your brain. And there are other complications, for example, in your liver. So people with diabetes are also at high risk of getting non-alcoholic fatty liver disease, so fatty liver or liver accumulation or fat accumulation in your liver, and also problems with your lungs. And with the arterial wall, so atherosclerosis also is often seen in people with diabetes. And they also have a high risk of cancer. And I think because you can imagine that these high glucose levels there, it's not just one organ that suffers from the high glucose levels, the glucose is everywhere in your body and this can damage the entire body actually. But then still we haven't given an answer to the question why the high glucose levels lead to cardiovascular disease or this cardiovascular mortality. And actually we don't really know. There are numbers of papers or research that tries to find out why people with diabetes have a high risk of cardiovascular disease, but it's not really known. There are some suggested pathways, and I will just go over them very quickly. So one of those is the oxidative stress. I already said that when you have a lot of glucose inside your body, you may imagine that it is not normal, that your body responds to this as well. We didn't expect this, what should we do with this? And this also one of the theories, I would say, that is proposed as an explanation for the relation between diabetes and cardiovascular disease. So here you see 4 very complicated pathways. You don't have to know them. But it's just to show you that, there's a lot of things happening in your body when there's a lot of glucose circling in your vascular system, and that all these pathways result in oxidative stress. And this is something that is thought to be related to vasoconstriction, inflammation, atherosclerosis, and by that causing cardiovascular disease. But of course, this is just a suggestion. There have been cell experiments that show that these pathways exist and that there is indeed oxidative stress occurring, but how this then leads to a cardiovascular event is not really known. Another theory is about the glycemic variability, because, I said that very high glucose levels in your body is not healthy, or that your body can exert stress from that. But there's also another theory that thinks, no, it's not just the hyperglycemia, it's actually the variability in glucose levels. So it's not so much a problem that your glucose levels stay high all day. It's more that it goes down and then up again and down and up again. And this theory, they say actually that it should be the normal range of fluctuations should be something like this, but when it's higher, so there are more going up and down and the ranges are higher than normal, that this is causing damage to your vascular system. But also for this, there is no proof that this is really the causal pathway. And then there are also other risk factors that are related to cardiovascular disease. So people with diabetes often have high lipid values, so high cholesterol levels, high triglycerides, and those are also related to the risk of cardiovascular disease. And the same holds for hypertension. Many people with diabetes also have high blood pressure, and obesity or being overweight is also something that often goes hand in hand with diabetes, especially type 2 diabetes. But still, what they showed in the Framingham study is that even if you correct for all these factors, people with diabetes still have a higher risk of getting cardiovascular disease. But yeah, we don't really know why that is the case. So if you take a closer look to what's actually happening in your vascular system, I show you here, the sugar cube should reflect something like your glucose, and you see also there's insulin because that's just circulating in your vascular system. And for this, I would like to ask you a question, because glucose is not only circulating in your body, but it's also stored in different organs. And I'm not sure if you thought of this before, but I would like you to think about the question, where is the largest glucose deposit in your body? So I put down 4 options here: your liver, your adipose tissue, your skeletal muscle, and your red blood cells. And I just want you to think about what would be the most logical option. Anyone who wants to share what he or she thinks? Who thinks? Yeah.
The liver.
Okay. You all agree with the liver? Yes. So actually, it's the skeletal muscle. You were close because there's a lot of glycogen stored in your liver indeed, but most is actually present in your muscle cells, which is actually quite convenient because if you start moving, you need the glucose as a fuel source, I would say, energy source. And it's very convenient if it's just closely located near the place you need the glucose. So there's only very limited amount stored in your adipose tissue, and 4 grams of glucose that is stored in your red blood cells refers to the HbA1c. But this is, well, compared to the muscle and the liver storage, only a very small amount. In this picture, you see how glucose and insulin should vary over the day. So it's not that your glucose levels, when you're healthy, just stay flat all day. They fluctuate as well, and the same holds for insulin. So every time you eat something, your glucose levels rise, your insulin levels rise, and then after a while, they go down again. And this is something that's happening in a lot of, throughout the day and every day again. And what you see here is that there's a difference in what people eat. So you may have heard before that when you eat something with simple carbohydrates, that your glucose levels rise very quick, whereas if you eat something with more complex carbohydrates, well, the rise is a bit going down. So that's actually what this figure shows you. But you see that the glucose and insulin is in blue, that they closely follow each other, which is actually how it should look like. But as I said, in people with diabetes, that is not the case. And I think to understand this better, it's good to take a closer look in how these two molecules, so insulin and glucose, relate to each other. So what's happening when you eat something? Well, your blood glucose levels rise, and your pancreas starts to produce insulin. So the beta cells of your pancreas produce insulin, and this makes it possible that your muscle or your adipose or your liver takes up the glucose that is circulating in your vascular system. And as a consequence, your glucose levels go down again because it's not any longer circulating, it's stored in any of these organs. And what happens when the blood glucose levels are too low? Well, your body senses, oh, we should have some glucose circulating because otherwise we will faint. So what's happening then is that glucagon, which is actually the kind of counter hormone of insulin, which is produced by the alpha cells in your pancreas, glucagon is produced, and by that the liver starts to break down the glycogen, and it's released in your blood vessels again. And this increases your blood glucose levels again, and you can imagine that this process is just going over and over again. But yes.
What would happen if we would eat all day long, like snacking all the time? Would the muscles just keep storing the glycogen or glucose, or would they stop when the blood sugar would stay high?
Yes, and that's also what we see in people with diabetes, for example. They just pee out the excessive glucose because if you cannot store it anymore, it should go somewhere, and that's why the sugar levels in the urine of people with diabetes are very high, for example. Yeah.
And is this in a person without diabetes? Okay, that's a bit off the topic, but is this unhealthy? If it's
you mean when you eat a lot of glucose the entire day? Or,
yeah.
Well, I wouldn't say that having very high glucose levels throughout the day is very healthy, but if you're healthy, I would say that your body will produce sufficient insulin to store it somewhere in your body. It's an interesting question, how kind of what the limit of your body is to another way that may also be what's happening when you eat a lot of glucose or carbohydrates is that when you start moving, your glucose levels also go down. So we also see in some studies that when people, when they eat a lot, they also tend to start moving again or start to move because their glucose levels are very high. So that's actually the same as when your muscle takes up the glucose. Also, when you're moving, your glucose levels go down and the circle starts over again. Take a closer look how this actually works. The question is, what does this hormone insulin do? Well, a lot, I would say. This is a very overwhelming picture. I will guide you through it, but it's a cell. Here you see a nucleus, and this is the cell membrane. And you see here, there's an insulin receptor. And when insulin binds to this receptor, many processes are taking place inside the cell. not only related to glucose, but a lot of other things such as sodium transport or protein synthesis. So it's just to understand that when people are not responding well to insulin or not able to produce sufficient insulin, it's not only their glucose metabolism that is affected, but there are many more processes going on that may be disturbed. So I already said that when insulin binds, well, something is also happening with glucose, and for that I think it's better to take a closer look. So we actually also refer to the insulin-dependent glucose uptake, and that means that the uptake of glucose can only happen when insulin binds to the cell. So what you see here, if there is no insulin bound to the insulin receptor, glucose just travels throughout the blood actually and does not enter the cell because there's no transporter that can move the glucose from outside the cell to inside the cell. This can only happen when insulin binds to the receptor. So what you see here is that when insulin binds, there's this whole signaling cascade going on, and this makes sure that the blood-4 transporter moves to the cell membrane, and only then glucose can enter the cell. So you can imagine that if there's no insulin present, that your glucose just stays in your bloodstream and it won't enter the cell. And this is actually why people with type 1 diabetes, for example, because they don't produce insulin, have very high blood glucose levels. And the glucose does not enter their cells. Besides these insulin-dependent way of glucose uptake, I just thought about the exercise or moving. Exercise is another way of taking up glucose inside your cells, because we know that when, well, also exercise causes this blood-4 transporter to move to the cell membrane, which is another way of getting the glucose inside the cells. And in type 2 diabetes, there is insulin present in the body because usually people with type 2 diabetes are able to produce some levels of insulin. But it's just that there's nothing happening inside the cell. So something is happening here with the signaling cascade, and therefore this transporter does not move to the cell membrane. And also then the glucose, of course, cannot enter the cell and it stays in your bloodstream, causing hyperglycemia. So there's this whole cascade going on, and we also don't really know how exactly or what is going wrong in the cells with people with diabetes. So what specific pathways inside the cell are not functioning. But we know that the insulin binds to the receptor, but just then there's nothing happening with the glucose transporter. And therefore also people with type 2 diabetes have high glucose levels. So this was the first part. I'm not sure if you have any questions so far. No? Okay. Then, well, I just said you can measure the glucose that is present in your blood, of course, but there are more ways of measuring glucose and also insulin sensitivity. So we already discussed this fasting plasma glucose or the 2-hour plasma glucose, and also a bit about the HbA1c. To share a bit more about fasting plasma glucose, it's often measured with a finger prick. Maybe you already did that also for a course here.